Hello friends, today we are going to discuss one of the important molecular technique used in molecular biology, that is Western blotting. You know, Western blotting. In the previous classes, we have discussed at length about different blotting techniques. Say for instance, Southern blotting technique for DNA, Northern blotting technique for RNA, dot blotting technique for DNA and RNA. The essence of this blotting technique, that means the primary objective of this blotting technique is to immobilize a specific nucleic acid on a solid surface, say for instance the nitrocellulose membrane or an NR membrane, and probing the same by a DNA probe through hybridization experiment, that means hybridization what we call, following by the hardware radiography for its detection. That means in order to identify a specific target DNA fragment in thousands of pools of molecules, blotting technique plays an important role. So that means it is an essential tool in DNA analysis experiments. Just like the Southern blotting technique to dot blotting technique for nucleic acids, the Western blotting technique is for proteins. The concept is saying we have to immobilize a particular biomolecule on a solid surface, say nitrocellulose membrane or nylon membrane or even PVDF, polyvinylidene fluoride. Just identifying the target protein by a specific antibody. Just like we use the DNA probe with respect to Southern blotting in DNA, here we use specific antibody, what we call protein ligand interaction. Just like antigen antibody epitope paratope interaction in different varied immunological reactions, the same concept applies here. Let me work out here. Just like specific ligand interaction, that means protein ligand interaction, we can able to identify our target protein. Okay. First, we'll discuss the basic protocol. Say preparation of lysate, then the sample, followed by loading, then running of gel, then transfer to nitrocellulose membrane or PVDF, polyvinylidene fluoride membrane. Then visualization by particular antibodies and detective reagents. So it's about the basic protocol, how the Western blotting works. First we have to prepare the lysate. I will tell you what exactly the lysate means. We have to break down the concerned target component. For example, cell culture or a tissue, whatever the case, it is a cell culture. To prepare the lysate, that lysate, the word it implied, break down the lysis. Then the components coming out of breakdown is lysate. Then out of it we have to prepare sample to be loading, to be ready for the gel running. Then followed by the transfer to a nitrocellulose membrane or a PVDF. Then finally the visualization by specific antibodies and detective reagents. It is in a nutshell, it is a schematic representation of how the Western blotting works. Okay. Then regarding the specific details, before we go into the working mechanism of Western blotting, before we go into the working mechanism of Western blotting, we have to understand different chemicals which plays an important role in Western blotting. Say buffers and detective reagents. These are very important components of Western blotting. Buffers are very important. Start in a sequence we'll discuss. Okay. First, lysis buffers. Then the name it implied, they meant for lysis. That means simply say NP40, then RIPA. Lysis for breaking down. Okay. RIPA stands for radio immuno precipitation assay buffer. Generally, the chemical constant they may vary with respect to its concentration. Generally present, that means the constituents in both these, that means MP40 and RIPA, which are important lysis buffers, or say NaCl, tris HCl, protease inhibitors. Then followed by, I don't know, in a sequence we will discuss the loading buffers. Then the running buffers, say tris glycine STS. Then transfer buffers, they are of say wet and semi-dry. And finally blocking buffers, say bovine serum albumin. Bovine serum albumin. These are the different buffers which are used in Western blotting. The first, the lysis buffer, say NP40 or the RIPA, that means radio immunoprecipitation assay buffer. The chemical constituents are like NaCl, tris HCl, protease inhibitors, and others at varying concentration. And after we break down, we have to use the loading buffer. How to load exactly? That means we have to prepare the sample. We have to use the loading buffer. Then while we run the sample, that means what we get protein components over the gel, we have to use a running buffer. Say the tris glycine SGS. The SGS means sodium dodecyl sulfate, and the kind of gel we use is SDS page. Sodium dodecyl sulfate polyacrylamental electrophoresis. Then after that, with the transfer buffer, finally the blocking buffers. Blocking buffers means after we transfer the protein components over the nitrocellulose membrane or the polyvalent fluoride membrane, we have to block. Then add the appropriate solutions of primary antibodies, then secondary antibodies for visualization. Primary antibodies act like DNA probes in southern blotting. They are useful for probing the target proteins, whereas the molecular markers do also play an important role. Whereas the secondary markers, the secondary antibodies, which are conjugated, and they play an important role for visualization along with detective reagents, say through chemiluminescence or calorimetric detection. That means in the end we could able to visualize the targeted proteins. That is this is about the protocol and basics theme of how the Western blotting runs. Okay, it's about different buffers. Then the step by step, I told you the first step is lysate, preparation of lysate. Preparation of lysate. You know how it works? I told you already. The breakdown components. Say you can use the cell culture, you can use the tissue, whatever the case it is. Here, for example, we'll take cell culture. Cell culture. Subject to lysate. How? We place the cell culture on ice. Then subject the same, that means cool wash, what we call cool wash by PBS, phosphate buffer saline. Phosphate buffer saline. After that, we have to subject the same to lysis buffers. That means after washing, what we call. That means washing the sense we have to suck off remove the PBS component. What we call aspirate. Aspirate PBS. Then we add lysis buffer at the rate of 1 ml per 10 to the power of 7 cells, for instance. You know, the say NP40, RIPA, radioimmunoprecipitation as a buffer. That's his buffer. You know, what exactly, the name it, they will break down. And the thing is, the cells will adhere to the dish. That means, just like, you know, tightly, just like gum, they adhere to the dish, and we have to use a cool plastic cell scraper. The cell scraper will scrap down the cells, and the cell suspension will gently transfer to a microcentrifuge tube. You know, lysis buffer, then scrapping with the help of a cool plastic cell scraper. This is very important. Then subject the same after we get in the microcentrifuge. That means put down in microcentrifuge tube. Microcentrifuge tube. Now after that, subject the same to constant agitation for 30 minutes at 4 degrees centigrade. Constant agitation for 30 minutes at 4 degrees centigrade. Then centrifuge. This is very important. Then at generally, you know, the centrifuge depends on the kind of the cell. Say for example, leukocyte require light centrifugation. That means the centrifugal force and the centrifugal time depends on the kind of cell. In general, what we say, the 20 minutes for 20 minutes and 12,000 RPM. Okay. After centrifuge, we have to aspirate the supernatant. Aspirate the supernatant and discard the pellets. This is very important. This is called cell lysate. This is an overview of how the thing should end. So that means we got this lysate preparation. You get the cell culture, say animal cell culture, present on ice, then use the PBS phosphate buffer saline, then suck off, that means aspirate the same, then use lysis buffer like NB40 or radio immunoprecipitation assay buffer, RIPA buffer. Then after that we'll get the cell scraper. The cell scraper who have to, you know, scrap off the cell off the dish, then gently transfer, that means the cell suspension to a microcentrifuge tube, then subject to constant agitation for 30 minutes at 4 degrees centigrade, then transfer the same to that of centrifugation at the rate of 20 minutes for 12,000 RPM, but it is subjected to kind of cells. Say for leukocytes, then a light centrifugation is required. So depending on it, okay, kind of. Then after that we aspirate the suspension. No, sorry, supernatant. This is called lysate preparation to discard the remaining pellets. That means at the end we got the lysate. It is either from cell or tissue. It is with respect to cell culture. Okay. Another thing is here we can use trypsinization. You know, after we subject the same trypsinization. Before we transfer to the microcentrifuge tube, an alternate route is also there. Before we transfer microcentrifuge tube, trypsinization followed by PBS. Trypsinization also PBS. It is also one of the alternate route for this particular step. Okay, this is about lysate preparation. And so at the end we got the lysate. Next step is sample preparation. You know, this is very important. The sample preparation includes sample preparation. Say for instance, this is a centrifuge tube. Okay. A small quantity will be used for protein quantification. To determine the amount of protein that we are going to use for gel analysis. Amount of protein we have to determine, then appropriate, that means equal amount of loading buffer have to use. Determine the determine the amount of protein plus loading buffer. So I prepare the sample. That means ready for gel analysis. The third step, what we call gel running. How? See, equal amounts of proteins has to be loaded onto the SDS page gels. Equal amounts of proteins. Equal amounts of proteins. Subjected to SDS page gel. This is very important. And after that, that means, say for example, say 20 to 30 mu g of total proteins of cell lysate. You know, we got the lysate now. Say out of total protein, 20 to 30 mu g of protein will be used in, that means to be allowed to the SDS page well. Then we have to run the gel. Run the gel. Running. Say for 1 to 2 hours at the rate of 100 volts. This is called running the gel. So allowing that means we have transfer, that means the sample has been subjected to the well SDS page gel and allowing the same for 1 to 2 volts, at 1 to 2 hours at 100 volts. You know, electrophoresis, simply the word is implied, the separation of the biomolecules based on its density. That's it. That means, say, when the current passes appropriately, the biomolecule will separate as with respect to the density. That's it. The basic theme of electrophoresis. The same will apply. Okay, we'll draw a diagram for better understanding. And then we have run the gel. Then the most vital step, the fourth step, is transferring the protein components to nitrocellulose membrane. You know, the fourth step, transfer to nitrocellulose or polyvinylidene fluoride membrane. Fluoride membrane. You know, we have used different things. They are like loading buffer, then running buffer. Here, the help of transfer buffer will transfer the same concept with the help of electric current. With the help of electric current, they transfer to the nitrocellulose membrane, that means the solid membrane, or PVDF, polyvinylidene fluoride membrane. For example, you know how it went. Say these are the, you know, the rods. Then this is how they travel. This is current, you know. The gel membrane setup. Say when you allow the current, say these are the rod, these are sponges, these are the filter papers, these are also filter papers, and this is membrane, and this is gel. This is a basic setup. How just simply they transfer from the gel to membrane. That's it. That's it. The membrane may be, you know, nitrocellulose membrane or polymethylated chloride. This is how they transfer through transfer buffers. Okay, then we got it. That means the membrane. I will tell you how the issue ends in the next step. What we call. So at the end, the protein content has been transferred to the membranes. Okay. The last step, what we call specific antibodies staining. That means protein ligand interaction. This how this ends. After we get the first we have to bring after we get the proteins on the membrane, we have to blocking the membrane through blocking buffers. Say I go to bovine cilium RNA, for example. Blocking the buffer. Say for instance, it will be by say overnight for food at the rate of 440 centigrade or room temperature per hour. It may be in 2 ways. Say 1 hour at room temperature or 4 degrees centigrade for overnight. This is generally recommendable. And after that, after we block, we have to add appropriate solution of primary antibodies, followed by the primary antibodies. What we call ligand protein interaction. They'll bind specifically to the target proteins. Then after that what we call probing. Simply it is called, this step is called probing. This is done, the primary antibodies. Then you wash the same with the appropriate solution, then add secondary antibodies. Secondary antibodies. That means the detective reagents we also have to add here. How we have to visualize. Say here visualization is with 2 ways here. Chemiluminescence by appropriate technique, luminescence, or calorimeter detection. Calorimeter detection. So at the end we would able to visualize. That means the primary antibodies are useful to bind the target protein, whereas secondary antibodies they be helpful for the visualization. And how it ends, say we have to draw say a simple representation with regard to the things, say what we got and how we could be able to visualize. Say this is a nitrocellulose membrane, this is a nitrocellulose membrane or a PVDF. Say measured in kilodaltons, and this is 20 and 30, 40, 50 and 60. This is line 1, this is line 2 and line 3 of different sources. It may be of X, say yeast cell extract, for example. This may be some HeLa cells, or some say mouse tissue. Different sources, okay? And how they appear, say like this, like dance. Just for understanding, I would like to draw like this. Is how so beta actin is one of the antibody used here. Okay, that means this is one of the simple representation of how on a solid surface we are able to identify. That means we are able to visualize the target proteins. This is how we can able to identify. Then and place which this Western blotting plays an important role in gene manipulation experiments. Gene manipulation experiments. That means through DNA analysis. this is an essential technique, particularly in gene manipulation. So the first step, the lysate, then you prepare the sample, then after that you prepare the sample on the, that means running the gel, then transfer the sample, that means the protein what we got, to a nitrocellulose membrane or polyimidine chloride, that means to a cell membrane, to a solid membrane, then alloy again electric current, and then followed by we have to add primary antibodies, then secondary primary antibodies for probing, then secondary antibodies here for visualization purpose. Simple. The lysate, the sample, the gel transfer, current, that means to around the current, then transfer primary antibodies, secondary antibody, visualization. That's it. It's about the basic technique, the working mechanism of plastic blotting. Okay? And thanks for watching. And next class we're going to discuss about other issues. Okay? Thanks for watching.